Assalamu alaikum. Hi everyone. This is section number 2 for Principle of Genetic Student Level 1, which is titled with Cell Division or Mitosis or Mitotic Division. And the main topic we'll talk about this section is about cell cycle and its phases and questions. All of you have studied before the type of cells and you know the differences between the plant and animal cells, and if you didn't remember that, please revise that before completing our section 2. Cell cycle. You have to know what does the cell cycle mean, and the cell cycle means actually that each eukaryotic cell has a repeating set of events. So I have a lot of events, and those events has repeating more and more and more, making the life of this cell, which is called the cell life cycle or the cell cycle. But you know that what is referred to? That referred to mitosis or mitotic division. Oh, that's our topic today. Yeah, but it's a small scale during the life cycle of the cell or the cell cycle. But the large scale or the large indivision or not division set or not division phase in the cell cycle called interphase, and that interphase is very important. Phase is not division phase, but it's so important. Why the interphase is so important in the cell cycle? Because without the interphase, mitosis not exist or not happen. Why is that? Because of the interphase is the preparations or all the needed for the mitosis to occur or to happen actually in the cell cycle, as we will see after a few minutes. You have to know that cell division is karyokinesis plus cytokinesis all together. That is interphase. Let's focus on the large scale not division stage on the cell cycle, which is called interphase. I told you before that is important and very important stage in the cell cycle because it help providing all the needed for mitosis to occur or to happen and in the cell cycle. And that interphase actually divided to three main steps or three phases or stages, as G1 for the first gap is for synthesis of DNA or replication stage, and G2 which is the second gap. And we'll learn together all the function of those steps. Okay, G1 or first gap, and G1 or first gap is the first stage in the interphase stage, as in not division stage. Okay, but actually before I started with explaining the G1 phase or first gap phase, I have to tell you that if you want to understand the mitosis or meiosis, just answer three questions: what, where, and when. Those three W's questions are very, very important to understand what's meant by mitosis and meiosis or mitotic and meiotic division. Okay, so let's answer our first question. What is the mitosis? Mitosis, which is you have a mother cell and its genetic material is 2N, so that you have a copy from mother and copy from father, so that mother cell has to produce two identical cells, and those two identical cells have to be the same genetic material of that mother cell. So I mean that all of them has 2N as genetic material. Yes, I mean that. So imagine that I have two oranges and I'll give two persons, each of them will take from me two oranges. Is it logic? Is not logic. So that I have to make some modifying of some modifying of this stage to be able to produce those identical cells. Let's know how that will happen. Okay, and the question number two. Question number two is, where does it happen? Mitotic division just happen in the somatic cell. It's happening in the somatic. As we said, mitosis undergo in somatic cell. Only on somatic cell? Yeah, in somatic cell. In your body, you have two types of cells, somatic and gametic cell or germline cell, but the mitosis undergo actually only in somatic cells, just like stem cells, epithelial, and all your body cells, excepting what? Excepting the gametic cells. There is no relation between mitosis and the production of gametes, as we will know or we will study together the next section. Okay. The question number three: when does it happen? When does mitosis happen? Mitosis happens actually all the time. All the time. What do you mean with all the time? It's all the time actually, because I need mitosis for growth. I need mitosis for repair tissues, or plant need mitosis for growth and reproducing. Okay, and I need, so the cell or the organisms need mitosis all the time of its life. Yes, it needs mitosis all the time, or all the life cycle, or sorry, all the lifespan of the organisms. Okay, so mitosis happen all the time until what? Until death. Until death. Okay, we answered the 3W questions. So let's complete our section or our slide as we said, or as we told. G1, or first gap, I have to or I need a lot of cells, so I have to increase in size, increase in number, right? So that stage when the cell grows and performs its normal function, small cells became larger than and more than in the number. S phase, or DNA synthesis phase, or replication phase. What does that mean? What about this phase? At first you have to know that how many copies of chromosomes do you have? Yeah, sure. You have 2 copies of each chromosome you have. You have 2 copies of chromosome number 1, so that you take a copy from your mom and copy from your dad. So you have 2 copies of each chromosome. Also I have 2 copies of each chromosome, also XX chromosomes. I have 2 copies of X chromosome, but the male have copy of chromosome X and copy of chromosome Y. But let's return to that point, which is the copy of one chromosome is at this form. And this form actually is about 1 chromatid. So the chromosome at the normal way, at the normal phase, is 1 chromatid. Once it entered the S phase, look at this, after the S phase, it becomes at this form. What happened? It's duplicated from 1 chromatid to having 2 chromatids held together at that point, which is called centromere. So let's revise that again. The chromosome enter the S phase from the G1 phase, and the chromosome shape in the G1 phase is that shape. Actually, it's 1 chromatid. That's called 1 chromatid. It has a small arm and a long arm, or short and tall arm with a centromere. Okay, once it entered the S phase or the synthesis phase, so that we call the phase the DNA synthesis. Okay, once it entered the S phase, it became at this form, which is mean it duplicated from one chromatid to two chromatids. Okay, and the two chromatids are connected together with the point called centromere. Okay, well, so I understood that now. Sorry, I understand that now, why the mother cell could give two identical cells, so that it divided it or replicated at the S phase. Okay, the stage number 3 in the interphase is G2 or second gap, and the G2 is the period between the S phase during the life cycle of the cell or the cell cycle. It's a gap between the S phase and mitosis. So I'm close to mitosis now. So what about mitosis? Mitosis, okay, let's say that again. It's two end cells and the mother cell have to produce two identical cells. Okay, and what is the cell? The cell is about nucleus covered or enclosed with a membrane, nucleus membrane, and there's some organelles, cytoplasm, and whatever, a lot of organelles, right? That so those organelles are divided also in the S phase, and the cells became larger than the normal. And I have some materials to make the copies, or to make to make the mitosis occur. I have some proteins, I have some RNAs, I have some enzymes, I have something just like you have to travel. You couldn't travel right now. You have to prepare your passport. You have to prepare all of you needed for that traveling, right? You have to make your luggage and so on, so that I have some ingredient to make my soup. I have to get my ingredient to make the soup. Okay, so I have something must happen in the G2 phase or the second gap to be ready to enter the mitosis or the mitotic division. Okay, so we right now entered mitosis, so I have to know what the mitosis phase is. Mitosis divided into 4 phases, and those 4 phases called number 1 prophase, number 2 metaphase, number 3 anaphase, and the last one is telophase. What's happened in all of those phases? Prophase, if you remember, is the first stage in mitosis or in the life cycle. No, it's the first divided phases in the mitosis. Remember that all of the previous stages are not divided stages. It's just stages non-dividing from the interphase. Okay, but the first dividing cell phase is prophase. In prophase, the cell begins the process of division, and actually, as we will see, the chromosomes, and the chromosomes is the form which is genetic materialis. Okay, the chromosomes condensing together, the protein attached to the DNA, because the chromosomes to go from long thin structure to short fat one, which makes them easier to pull apart. we need to agree that every stage will do its function and transfer the product or the end product of its function to the next stage, so that in the prophase, the chromosome, I said that the chromosome will be condensing or will condensing, right? The protein attached to the DNA causing the chromosomes go from long thin structure to short fat, which is mean it is so like a cucumber, as cool as cucumber. It's in the resting stage. That's the resting stage. There is nothing to do, but there is an order now to make the mitosis, and that order make the cell has or the urine material or genetic material, which is chromosome, condensing from the resting net, from the resting material, to be the form of chromosomes, just like this, The main events which occur in the prophase is that the nuclear envelope disappears. You have to know that the nucleolus have an envelope, right? The nucleolus has an envelope, so that in the prophase, the nuclear envelope disappears. Okay, the spindle fibers start to form. What that spindle fibers? I know its function in the next stage. Okay, growing out of the centrosomes toward the chromosomes. What that? The spindle fibers. Okay, that is the early prophase. I can see the nucleolus. Okay, but late prophase I can't see it. Okay, metaphase. Metaphase is the second stage in the mitosis. Metaphase is a short resting period where the chromosomes are lined up on the equator of the cell. Look at this. Chromosomes. Oh, in the last stage, I couldn't see that form of chromosomes or that form of genetic material. But that form, or without making that form, the chromosomes can't stopping or can't line up in the equator of the cell at this shape. So that at the last stage, or at the first stage, which is prophase, the genetic material became that form, or the chromosome form, instead of the net, or the chromosome net. Okay, and okay, what happened right now? The chromosome lined up on the equator of the cell, and what? Spindle fibers, which we talked about in the last stage, or the previous stage, which is starting the formation from the pools here, will send its fiber, to attach to a specific pore here on the chromosome called kinetochore, and on the centromere called kinetochore. So the spindle fiber make its fibers to attach with the centromere at point called kinetochore. For what? For make the next stage able to do its function too, right? Anaphase, or the third stage of the mitotic or mitosis stages. Okay, if you remember that in the last stage, I mean in the metaphase stage. In the metaphase stage, the chromosomes lined up on the equator of the cell, and something called spindle fiber sends its fiber to attach with the centromere, especially in a point called kinetochore, and it is ready now to separate in the anaphase. Okay, so that they lined up and be ready to separate in the anaphase, lined up in the metaphase, and the separation will occur in the anaphase. Okay, and now look at this. In anaphase, there is two pools of the cells, and there is two groups of cells, two groups of chromosomes in each pool. Write that. But each of them is chromosome with one chromatid, so that it's the same content of the mother cell which we began with, right? Is chromosome with one chromatid, only one chromatid in each pool. Okay, so in anaphase stage, chromosomes separated at the kinetochore and move to up the pools of the cell. Motion results from the combination of kinetochore movement along the spindle fibers and through the physical interaction of polar microtubules. So here the cell depending on, or the separation depending on the force of the spindle fiber in separation. Okay, after that, sorry, this is another image for anaphase. Look at this. They say no, you never said there was a second anaphase. Okay, actually it is a picture for meiosis, but the same is the same idea or the same way. These are the same copy of chromosomes, homologous chromosomes. They like sister chromosomes, right? They were connected together at the same centromere, but the force, according to the force of microtubules, okay, they separating from each other. Okay, each of them go to another pool to her sister. Okay. Okay, that the fourth stage and the last stage actually in the mitosis. In telophase, the cell actually divided. The chromosomes are at the pools of the spindle. You look for this. Look for this. The chromosomes became in a resting stage again. That is a chromatin net, not the chromosome form, because it ends its function, and the nuclear envelope reforms. Nuclear envelope which is disappeared in the first stage in prophase. It's done now, so it reforms again. Okay, and the spindle fiber, I need that now? No, it's done for it, so it disintegrates too. So the chromosome or the genetic material in in telophase became in this form actually. So I have two identical nucleus in the same cell. Until now, they are in the same cell. Okay. Okay. Let me remember that with you. The cell division is karyokinesis plus cytokinesis, right? We agreed with that in the beginning. So the cytokinesis is the second part of the cell division. So the cytokinesis, I mean that the organelles also get divided up into the two daughter cells. Okay, what that mean? I mean that cell division consisting of two parts: nucleus division, or what we called karyokinesis, which is genetic material divided into two identical cells. Okay, and also another part of the cell division is the cytokinesis, and the organelles also have to divide it up into the two daughter cells. okay? And that's what happened actually in this stage. But there is a small problem that plant and animal cell actually they are totally different. So also the cytoplasm will divide in those cells in different ways. Cytokinesis. Cytokinesis in plant cell. We agreed that cytokinesis in plant and animal cell will be totally different according to the the formation of each cell of the two cells or the two types of cells. Okay, cytokinesis in plant cell actually a new cell wall made of cellulose or lignin or pectin or hemicellulose, actually from the same made of the same material of the mother cell. Okay, and that will form from the same material or from this material as for example as cellulose. It make a separation wall between the cell until the last stage of mitosis or the telophase. The two identical nucleus or the two identical cells is existing in the same cell, right? We have to separate it to achieve the target of mitosis, that 2N make two identical cells, each of them has 2 and 2. Okay, so it will be make a formation like this from the enter or from inside to outside the cell, right? This like this, like this, and it completed totally, and then separate the two identical cells each from them. But in animal cell has another shape from the plant cell, so it makes the separation or make that action in another way, which is like this, just like a balloon, and you make stress from the like this from the it's called cleavage. It means from the middle of this balloon I make stress and separating or pinching the cell in half. Okay, just like this. Okay, it also ring of actin fibers forms around the cell equator like this, and as we said, pinching the cell in half. So that let me revise the stages of mitosis again. This and the first time I will say that is that prophase? No, it isn't prophase. It's just like interphase or pre-prophase. Okay, but this is prophase. Why I call that prophase? Why I call that prophase? Because in the prophase the chromosomes or the chromatin net became in the chromosome shape. To make what? To line up in the metaphase in the equator of the cell, to attach it by the spindle fibers in the kinetochore on the centromere position, to separate in the anaphase, right, like this, and to form two identical cells inside the same cell in the telophase, right? That, okay, that's an animation, and that's the live cells. Okay, that's interphase, and this actually the chromatin net or the prophase. It doesn't like the same chromosomes as here, right? And that the metaphase or the chromosomes lined up the equator of the cell, and this the anaphase, which is chromosomes separated, each chromosome from two chromatid So one chromosome in each pole with one chromatid, and this is the telophase, and it became a nucleus again, and it will reform the nuclear envelope and reform the nucleolus, and also the spindle fiber disappeared, and this a plant cell, so I can see the wall forming here to be ready to cytokinesis step. Okay, that's another one. This is what I can see chromosomes over the cell, or I can see chromosomes lined up the equator of the cell. Yeah, they're lined up in the equator of the cell, so that phase is right, metaphase. And number two is just like fingers. It's just like fingers, and all of them separating in each pool, in another pool. So that anaphase, right? And this prophase, and these two nucleuses inside the same cell, so that it right, telophase. And those our references for our section today. I hope that everything is clear. If there is some problem for you, if there's some problem with you, or some problem with understanding anything, please note that, and we will discuss during our section. And see you next video, inshallah.